Hi everyone, welcome once again to another tutorial session in Python for bioinformatics. In today's video, I am going to show you how you can use Python codes to find the complements of nucleotide sequences. Let me also say that the notebook for this tutorial is available on my GitHub page. The link is in the description box. Now, in order to find the complement of a sequence, you need to first obtain that sequence or sequences, and then you create a dictionary object to store each of the 4 bases and their respective complements. You then use loops to iterate over the bases in the sequences, find their respective complements, and combine them. It's very simple to do. Let me show you how. So we begin our activity by handling a single sequence. So that means we have to obtain our sequence. So this is the sequence. I am going to paste this in the description box so that you can use that to feed Python. Now, you can also generate your own sequences using Python, and I have a video that shows you how to do it. The link is in the description box. You can also use sequences from FASTA files or GenBank files. I have separate videos that shows how you can use Python to access these files. The links are in the description box. Now, let's create the sequence. We have created it, so let's just print it. And we have it here. If you also want to find the length of the sequence, we can use this simple command here. We can say len DNA, and we have it here. So we have 15 bases here. Let's move on to the next step. So the next step is to create a dictionary to store the bases and their respective complements. Dictionaries comprise of key value pairs. And so with dictionaries, if you want to have access to a value, you call the key. I'll show you how we create it here. We are going to include the name as well. That is the name for that dictionary. So we say base complements, because the information we are storing is about the complements for those 4 bases. So to create a dictionary, we use this. That is the curly brackets. Let's do it well. So we have our curly brackets here, and then in the curly brackets, we specify the key value pair. So the key will be the base, and the value will be its complement. So let's start with A. So the complement for A is T. So we give the key, which is A, and the value here is what? T. So for each key value pair, we separate them with a colon. So the left here is a key, here is value. So let's move on to the next one. We separate with a comma. There's another one. So the T has its complement as A. So we have it here as well. So the key value here, we separate them with a colon. And then for each key value pair here, this group here, we separate them from other key value pairs using the comma here. That is how we do it. Let's do for what? C. The complement is, and then for G is what? C. So we have them here. So we have the dictionary objects created here, and we are saying what is base complements, because the information is about the bases and their respective complements. So we have it there. let's just print it. And we have it here. Now, before you proceed, when you have your dictionary, you can make queries. So, for example, if you want to answer this question, what is the complement of A? Then, what you do is that you call the dictionary. And then you indicate a square bracket. And then in the square brackets, you put in your query, which is A. Because the question is, what is the complement of A? So A becomes our key. So once you put the key here and you run, the value that was assigned to this key will be displayed. So complement of A is T. The same thing, what is the complement of, let's say, C? Let's use C. We say base complements. And we give the nucleotide. And it's what's G. So using these steps, you will now expand it and then do it for the entire sequence here. So let's move on. So now we answer this question. Find the complements of the sequence. Let me add this here. We are looking for the complements of this sequence here. So we begin. We have already created that sequence here, which is the first step. And then we've also created a dictionary that contains information, that contains the complements for each of these bases. So we move on. So here we have approaches we can use. This is one of the approaches. I will show you two approaches here. This is approach 1. And here we use the traditional for loop. So let me show you how we do it. If you are using a traditional for loop, you first need to create an empty string. So we say, let's say sequence complements. And then we create the empty string here. Then we now use the for loop to iterate over the sequences. This sequence here, we iterate and get the bases, each of the bases. And this is how we do it. So we say for base in what's DNA, because we gave the variable name as what's DNA. That's what we are doing here. So for base in DNA, we now say base complement, because we want the complement for this base. And this is where the dictionary is coming. So we call the dictionary base complements. And then we add our base of interest, which is base. We are iterating, so we use this here. Let me also chip in that I have a separate video on for loops, so you can also watch that to try and understand how for loops can be used to make your life easier. So we have this here. So this means that when we call that base, its complement will be pushed here. After that, we now add the complements to this string here. We push them here and we gradually elongate it. So we see sequence complements plus equals base complement. So that is all. We now run it. So now we have the complements generated. So let's print it. Sequence complements. So we have it here. But of course we can print both the original and then the complement so that we can easily compare them. So let's print both. Let's say print what? Sequence. Let's use this for the original sequence here. And then we indicate the sequence here. And then let's print the complements. Let's say sequence complements. And we add what? The sequence complements. So let's run it and then compare. So now we have them here. So the original sequence is here, and we have the sequence complements. Let's make this better. Let's align them properly. I'm just adding some space characters here. So we run it, so now they can be compared easily. So A is what? T. T is A. C is G. So you notice how Python has done this nicely for us. It was very quick and very easy. And that is the power of programming. Because with programming, you can analyze and process sequences that are long. And that is something that if you are doing manually, it's close to impossible. So this is for the first approach. Let's look at the second approach. So in approach 2, we use list comprehensions. List comprehensions combine the traditional loops into a single line, and it's relatively faster. So if you are using or if you are processing several sequences, then you can choose to use list comprehensions. So this is how we use it. It's a one-liner. So we say sequence complements. Now let's differentiate this from the first approach. So let's say sequence complements 2. Let me do it here. Let me enlarge it to make it better. Yes, so we have sequence complements 2. So we say we put the query or the information in a square brackets. That is how we use list comprehensions. So we call the base complements. And we indicate base. We are using loops here as well, but then we have a way of using the for loop here, and that is what we are doing. So we say base complements base for base in DNA. We are still using that same original sequence here, the DNA we created here. So we run it. We have our list comprehension here. Now it is done, so let's print it. I would like to show you something here. So print sequence complements 2. So when you use a list comprehension, this is what you get. But it will be better and nicer if you convert this to a string so that it's easy to understand and read. So we convert the lists to a string. And this is how we do it. We call, you see, sequence complements 2 equals empty string dot join, empty string dot join, and then we indicate the sequence complements 2 again. So this particular approach works if all the members in your list object are strings. And this is the case with our hot data here. And that is why we are using this. So let's run it and see what happens. Let's print it. So notice how that has also been done nicely for us. Now let's compare this sequence complement 2 to the first one. So let's print both. Let's do it here. We say print sequence complement. That is the first one. And then print sequence complement 2. And we have them here. So notice that they are the same. Okay, so we use different approaches, but the results we get will be the same as we've done here. So let's even add the original one here. So let's say print original sequence, that is DNA, and then let's print approach 1. That is what sequence complements. And then let's print approach 2, which is what the list comprehension. And that is sequence complements 2. So let's print them and then compare. Of course, here, let's make it nicer. Let's add some space here so that they are all aligned. But you don't have to add a space. It's just for me to easily compare them. So we have all of them here. So notice they are the same. We have the original sequence here. And these are the two approaches we use to find the sequence complements. And these are very cool activities you can do when starting your programming journey. So these are the two approaches to find the complements of nucleotide sequences. But then what we've done so far was to find the complements for a single sequence. So let's look at how we can use this approach we have here on multiple or several sequences. So to find the complements of multiple sequences, the approach we are going to use will combine functions and loops. So combining functions and loops helps us to process multiple sequences. And so it is important you know this. I have separate videos on functions and loops, and I'll leave those links in the description box. So first of all, we need to have our sequences. So we have 3 sequences here. I am going to leave this also in the description box so that I can use that in this tutorial. So let's create the sequences. We have 3 of them. Now there's a list object, and I have a separate video also on lists. You can also watch that to try and understand what lists can do and how you can use them to process sequences. So we have them here. Now, if let's say you didn't know how many sequences were present here, you could just use the len function to find the number of sequences here. So there's just one stuff I wanted you to know before we proceed. So we have it here. So what we are going to do is to create a function that will find the complement of a sequence, and then we reuse the function for all the other sequences. Okay, so with functions, it's easy to reuse the codes and apply them on other sequences. So that is why we create it. So again, I repeat that I have a separate video on the function, so you can watch that to understand what the codes we are going to use mean. So we create the function by issuing this keyword here, def, and then we give a name to the function. So let's say sequence complement finder. Notice the underscore here. Please make sure you also use them. So we have the name entered. Let's add a parameter, which is sequence. So this here will act as a placeholder for all sequences that we will feed into this function. Let's make it better here. So once we have the name created, we begin by first creating the dictionary objects, the one that contains information of the bases. And the information is what? The complements for each of those bases. So let's see base complements. Now we know how to create it, so let's do it quickly. So we have the function created. You should also cross-check to make sure that the syntax is correct. So that is what I'm doing right now. Now it's fine. So what do we do next? Let's give it a name. Let's say sequence complements, because that will be the next step to find the complements. So we are going to use list comprehension here to make it faster for us. So we say base complements base for base in sequence. So this is the sequence. So we have it there. After that, we convert it to a string. So we see sequence complement equals empty string dot join sequence complements. Now, when we have variables in a function, those variables or values are accessible only within that function. They cannot be accessed outside the function. That is how Python has been set. That is the default setting. So in order to access a value here, there are a number of approaches, and one of them is to use the return keyword. So we are going to return what the sequence complements. And so this means that when we call this function, we assign to a variable so that the return statement here will push the result to that variable. So we create that function. Now we have created a function, we have all that we need. So let's create it. Now we have created it and we are ready to go. So the next thing to do is to use a for loop to iterate over the list of sequences, these ones here. And then when we iterate for each of those sequences, we call the sequence complement finder, and this will find a complement for us. So let me show you how we do it. Let me just add this comment here. Okay, so now we have it here. So let's call the for loop. So we see for sequence in DNA sequences. We are using this because that is the name here, the variable name here. So that is why we are using it. So we call that and we see sequence complements equals, and we call the function here sequence complement finder, and we feed it with the sequence here. Then we are going to print the original sequence. Let's print the original sequence here. Let's add the sequence. Then we print what? The complements. And that will be what? Sequence complements. So we just want to iterate, find their complements, and then just print them. So we have it here. So let's do it. Let's issue this command. So nice. It has been done for us as we expected. Let's just format this properly so that they are all aligned. So let's rerun it. Good. So now we have them here. So you can compare. We have the original sequence for each of them, and then we have its what complement also here. So that is very, very cool here. ATCTAG and all that. So this is how we do it when we have multiple sequences. So using functions and loops, we can do so much with Python. So here, if let's say you want to maybe have access to those complement sequences later, you can also create a list, and then in the list you append all those complements that you have. So let me just show you that one here. So we can just create an empty list. Let me show you that one quickly before we go. You can see complement sequences, and we have this here. And then when we come here, we use this code here. We don't need this again. Then we see complement sequences dot append sequence complements. So let's bring it up here. So we have it here. So we create the empty list, and then after we are done with the operation, we append the value we get into this here. So let's run it. So we have it done. So we can just print that list. And we are going to have what's there. Complements also listed here. We can print the original one, which was what DNA sequences. And then we print the complement, which is what? Complement sequences. And now we have them here. So now we know the original, we know what's the complement. We can also make this nicer by saying what? Original, and then what? Complements. And then we run it. So now we have them here as well. We can just make it better. And now we have all of them here. So you know that for each sequence here, we have its complement also displayed here. So these are things you can do along the way with Python. So, and that's it. So thanks for watching this video, and I believe it's been very helpful for you to learn as part of your Python programming lessons. So in the next video, we are going to look at how to find the reverse complement of sequences, and I believe you are also going to enjoy that as well. So I'll say thanks for watching this video, and I'll see you in the next session. Goodbye.